The medications and the formulations that we're working on, they've got the potential to help millions of people. Before we get anywhere near any kind of living creatures, we work in cell-based assays, where we will identify a number of the drug parameters. So we'll work with different formulations that will have come through from our colleagues in chemistry, and we'll be looking for different parameters on those, such as if they're toxic or less toxic, or efficacy if they're working against viral targets. And then they'll feed into the in vivo work that's done within the group. We work with a large amount of drugs, looking at different diseases. COVID is one, but we also work on hepatitis C, malarials, TB drugs. And the bioanalytical quantification is taking all samples that are generated in vitro or in vivo. We use high performance liquid chromatography or a liquid chromatography mass spectrometer. And essentially they're a balance, so they just quantify the drug, they just measure how much is there. The difficult part, and where I spend the vast majority of my time, is developing assays for these drugs. So when we get them, they're in what we call a matrix. So this could be a saline solution or plasma or tissue. The first step that we've got to do is separating the drug from this matrix so we can actually determine how much is in there. Then we have to do separation from what's in there, because no extraction method is completely 100% efficient. So you'll get some background noise, something that could interfere with the drug that you're looking for, so it could mask it. So for that, we use a technique called chromatography. And all that is doing is it's running your liquid sample through a column, and that column has got a solid phase inside it, and the drug has got an affinity for that solid phase, so it will interact with that and remove itself from the liquid. So you'd have your contaminant coming off, and then your drug would slowly come off as well. And that's where you can quantify it. Some of the drugs that we work with are very, very poorly water soluble, so you put it in and it'll just clump in the bottom, so it's not able to be used essentially biologically. The nanoformulation can hold the drugs in a suspension, so it can effectively carry more drug in that liquid, so it's not dissolved, but it's holding it in a suspension. Then also there's a number of different mechanisms that the drug doesn't take advantage of that the nanoformulation can. So there's a number of mechanisms that the cell will take up different natural vesicles. The nanoparticle are able to take advantage of these, so instead of it just passively entering the cell, it's an active mechanism. The cell's actively pulling more in. The biggest challenge with it is making sure that the assays are reproducible and sensitive. So we need to make sure that we're getting very low quantification on these drugs. Say we're looking at a nanoparticle that we want to deliver orally. We would look at running an in vitro assay using cells called CACO2s, which simulate the gut lining. And you would see how much drug is able to go through that lining, simulating how much would go through when you've taken it orally into your systemic circulation from your gut. So the nanoformulation will look at traversing that gut or taking advantage of specific mechanisms to make it cross that gut to increase how much drug is getting in the body. So then we can screen many, many different compounds, and we can look at whether they're able to perform better than the original drug. So our reformulation of it can then improve it. And then this has got potential benefits of making the drug more effective or making it more cost effective. Because if you can get more into the body, then you don't need to give as large a dose at first. So if you're given the traditional formulation and 10% gets into the body, 90% going out as waste. But if you can do a nano formulation that got to 50% or higher, then all of a sudden you've got a lot more drug that's able to get into the body, so you can save money. And that's something that's been quite important with a lot of the neglected diseases. So things like HIV, malaria, definitely the parts of the world that they're effective, that kind of saving would make a massive benefit.